Hi guys, Miss Francis here. So I'm a very visual person. We annotated images, we watched some video clips, and then we took some notes. But I would like to look at the Calvin cycle in a flow chart, highlighting some of the important events. So the Calvin cycle occurs in three phases: carbon fixation stage, reduction, and regeneration. Let's tackle carbon fixation first. So in carbon fixation, three one-carbon compounds, not necessarily sugar, because carbon dioxide is not sugar. But anyway, a one-carbon molecule joins with a five-carbon molecule, ribulose bisphosphate. These are the reactants during carbon fixation. Using the enzyme Ribisco, these molecules join to form a very unstable six-carbon molecule, which then immediately splits to three carbon molecules called phosphoglycerate, and we make six of those. So you need to know phosphoglycerate, which is also known as PGA. You need to know Ribisco, and you need to know ribulose bisphosphate. The next stage of the Calvin cycle, the next phase, is reduction. So I take those phosphoglycerate molecules that I got during carbon fixation and add a phosphate to them. Well, where do I get that phosphate from? I get that phosphate from ATP that was generated during the light reaction. When that phosphate is added, I form 1,3-biphosphoglycerate. That biphosphoglycerate is going to get reduced, meaning it gains an electron. Where does it get that electron? It gets that electron from NADPH, which came from the light reaction. So NADPH is going to get oxidized. It's going to lose an electron, thus reducing 1,3-biphosphoglycerate. And as a result of reduction, I get my first sugar. I get G3P, which is a three-carbon sugar. And I'm going to make six of these. Of those six, one is going to exit and be used by the plant for food. In other words, it's going to form glucose. The other five G3Ps, which are also known as P-gal, don't confuse that with PGA. So the G3P or P-gal gets recycled so that the Calvin cycle can occur all over again. So then this last stage of the Calvin cycle, or this last phase of the Calvin cycle, is regeneration. During regeneration, the whole point is to create Ribisco bisphosphate so that it can once again accept another carbon dioxide and continue the cycle. So how does that occur? Well, I have my five three-carbon molecules, my first sugar that I made, which is G3P, also known as P-gal. Because I have five of those three-carbon molecules, I have a total of 15 carbons. With the help of ATP that came from the light reaction, those molecules, those carbons, are going to be converted into three 5-carbon Ribisco bisphosphate. So I still have those 15 carbons. Originally, we started out with three carbon dioxides. Three carbon dioxides means this cycle spins 3 times. From those 3 times, I end up with one GP3. I end up with one 3-carbon molecule. In order to get glucose, which is a 6-carbon molecule, I have to do 6 spins. So ultimately, the Calvin cycle is 6 spins, which creates two GP3s, two 3-carbon molecules, which can then form glucose. So overall, I have 6 carbon dioxides to make 2 G3Ps, which will give me 1 glucose.